This is Rochelle, your Microbiology IT tutor. Welcome to today's Microbiology retention session where we will be studying translation and protein processing. If you need steady resources to help you get that A, then grab your Microbiology IT workbook. The link is in the description box. Translation and protein processing. Here we have 3 branches stemming from it, and we are going to tackle the first one. So the first one is genetic code. And from genetic code, we have 3 branches. We have, let's do universal here. Degenerate. Oopsie. And then wobble. Base pairing. Okay, so with universal, in most organisms, you have the triple codons on the messenger RNA. And then to generate, we have multiple codons on the code for that code for the same amino acid. So we're going to do multiple codons that can code for the same amino acid. Wobble base pairing kind of goes hand in hand with degenerate. So we are going to say that it is there's flexibility in the third codon position, that allows one messenger, or not messenger, transfer RNA to recognize more than one codon. Okay, so that is the genetic code. And we see here in our notes, genetic code, the triple codons are nearly universal, degenerate. Wobble base allows flexibility. You have the start codon, that's the AUG, that codes for methionine. And then we have the stop codons that are UAA, UAG, and UGA. Some other key facts that you need to know is tRNA charging. What is that? Well, we have the enzyme aminoacyl tRNA synthetase that ensures the correct amino acid is attached to the correct transfer RNA so that the transfer RNA can go to the ribosome and drop off that amino acid to create the polypeptide chain. And this whole process is the tRNA charging process is needed, or the ATP is required for this transfer RNA charging. Then we have our ribosome structure where prokaryote ribosomes are 70S, 50S plus 30S, and then we have eukaryotic ribosomes that are 80S, and that they are 60S plus 40S. In the ribosome structure, you have A, P, and E sites that are involved in the polypeptide chain process, the protein process. Translate. Our next steps, we're going to cover translation. So our next branch is translation. 3 branches stemming from translation, initiation, and 3 branches from that where we have small ribosome subunits, a subunit that binds to the messenger RNA. Then we have a start codon, which is the AUG that is recognized by transfer RNA. And then our last branch here with initiation is that the large ribosomal subunit assembles. Let's move it below. Assembles. Our next branch in translation is elongation. And we have four branching out of elongation. We have the A site, where the transfer RNA goes to this specific site, and it already has the amino acid attached to it. Then we have the P site, where the peptide bond is formed, and then E site, where the transfer RNA exits. Then we have some factors to consider in elongation. So we have EF2, EFG, and then GTP hydrolysis. Our last branch is termination. And we have 2 branches from that one. And we have polypeptide is released. Peptide is released. And then stop codons are red. So the stop codons are red, or the stop codon would be red, and that will release the polypeptide from this whole process. The protein folding and processing. The protein folding and processing. We have 3 branches coming from this one. So the first one is the primary to quaternary structures. So it's the structure of the protein folding, of the protein, which is the specific foldings. So primary to quaternary structures. And then we have denaturation. That's whether it's a pH or heat that alters the folding, the structure of the protein. And when that's altered, then that means that the function of it is changed or no longer can do what its function is because of the denaturation. And then our next one is our last one is post-translational modification. And we can go to our notes where we see on our second point, translational modifications include phosphorylation, glycosylation, and disulfide bonds. So those are our three branches: genetic code, translation, and protein folding and processing. So we're going to go over our last few slides. So we have three questions. Question number 1. The stop codons for translation are usually, what are they? And you can find these in your notes, text. Number 2, the ribosomal site that initially binds incoming charged tRNA is the blank site. Number 3, the ribosome recognizes the start codon blank on messenger RNA, which codes for the amino acid blank. Multiple choice questions. Question number 1. Which elongation factor is required to bring charged tRNA to the A site? And you would find these in the notes. Number 2. Which rRNA catalyzes peptide bond formation in prokaryotic ribosomes? So number 3, a mutation changes the start codon AUG to ACG. What is the most likely consequence? Fill in flow challenge. Complete the sequence of translation events. Initiation, complex forms. Then charged tRNA enters the blank site. Peptide bond forms at what site? Blank site. And ribosome translocates. Then empty tRNA exits at what site? Application case. So an antibiotic binds to the bacterial 30S subunit, preventing base pairing between messenger RNA and 16S rRNA. Explain how this affects translation. I'll see you in the next interactive tutoring session. Let's microbiology it.